Okay, it's 10 o'clock, so welcome everybody on the third installment of Polish NMR seminars. We stay in the topics of bio-NMR with Dr. Witold Andrałojć from Institute of Bioorganic Chemistry, Polish Academy of Sciences from Poznań, and he will talk today about NMR studies of non-canonical nucleic acid structures induced by specific binding of metal ions. So Witold, the floor is yours.
Okay, thank you, Mateusz, for the introduction. I will now share my screen. Can you see the presentation now?
Yes, we can.
Okay, great. So thank you again for the introduction. I represent the Institute of Bioorganic Chemistry of the Polish Academy of Science. And as Mateusz said, I will be talking about NMR structural studies of non-canonical nucleic acid structures, more specifically those induced by specific binding of metal ions. But before I start, a few words about our group, because as I understand, these seminars are an occasion for the NMR community to know each other better. So at the Institute of Bioorganic Chemistry, the NMR studies have a very, very long history, very long tradition. The NMR group changed names several times, but what didn't change was that it was always led by Professor Zofia Gdaniec, until she retired at the beginning of last year from full-time work. And at that time, I took over the department. Luckily, Professor Gdaniec is still helping us with several projects. But since last year, I'm leading the group. I took the research in a few new ways, but I will show you that during the presentation. But we're also continuing some older projects. What didn't change is our specialty. Since the very beginning, the NMR group at our institute was specialized in the study of nucleic acid molecules. And you might ask, what's so special about the structure of nucleic acids? Because everybody who doesn't work with them, when asked about nucleic acid structures, will probably picture something like this: a double helix. In the case of DNA, a so-called B-type double helix. And indeed, this is the most typical canonical nucleic acid structure, held together by Watson-Crick base pairs. And everybody usually says that the double helix is held together by hydrogen bonding between bases. But actually, equally important are other types of interactions, like stacking interactions between neighboring base pairs. These are electrostatic and van der Waals forces. What is also very, very important to hold this double helix together is how it minimizes the repulsive interactions that occur when the two DNA strands interact. You might ask, what are these repulsive interactions? Well, if we look at the covalent structure of a nucleic acid molecule, we will notice the phosphate groups between every single nucleotide unit. And at neutral pH, each one of them bears a single negative charge. So actually, this molecule is very, very intensely negatively charged. It's a polyanionic biomolecule. And to put two of these polyanionic molecules together, well, it creates a lot of electrostatic repulsion. And the whole system is very, very strongly negatively charged. So actually DNA molecules, and RNA as well, never exist in solution alone. They're always surrounded by positive ions that neutralize this charge. And as you can see, these negative charges are actually quite evenly spaced along the biomolecule, which means that it doesn't form, at least this kind of helix, any specific strong metal binding site. It's just negative charge evenly spaced. That's why the most important type of interaction is actually non-specific. Most of the metal ions that neutralize the charge of the biomolecule form the so-called ionic atmosphere. So these are just cations, most usually metal cations like sodium or magnesium cations that move alongside the biomolecule. And what we actually see is not site-specific binding, but rather a depletion of negative ions in its vicinity and an excess of positive ions. Of course, if something more interesting happens structurally, there is some place where the phosphate groups or other electronegative groups are more condensed, then we can have site-specific interactions. And they can be direct interactions between, let's say, carbonyl groups or the phosphate groups with the metal, but they can also be mediated by hydration water, sometimes even very, very indirectly. And if we start to think how many of the two binding sites we have, we can look at crystal structures of large RNAs. This is an RNA that is several hundred nucleobase long. And we can see how many metal ions we can actually see in the crystal structure. These are all magnesium ions. And as you can see, there is around a dozen of them on a molecule that has several hundred nucleotides. So usually less than 10% of metal ions are site-specifically bound to nucleic acids, and only if they form some higher tertiary structure. Just a simple helix doesn't contain any strong metal binding sites. As you can see, these metal binding sites usually form where, for example, there's a turn of the backbone and several phosphate groups are located close in space. However, even these site-specific interactions aren't exactly strong if we compare them to proteins. And this is a point that I would like to illustrate with this table here. The dissociation constants of most of metal binding sites in DNA or RNA are actually millimolar, or in best case, micromolar. And probably this is actually the physical limit of what this biopolymer can achieve. And compared to proteins, where even for metal ions that are abundant in cells, we usually see nanomolar affinities. And for those metal ions that are very, very tightly regulated, we can even have atomolar dissociation constants. This puts us into perspective how weak the ability of DNA and RNA to site-specifically bind metal ions actually is. And this is a little bit of a paradox, that on one side, this is a highly negatively charged molecule. It needs these metal ions to form any kind of complex structure. But at the same time, the diversity of functional groups it can use to bind metal ions is so low that usually even the important, structurally and functionally important interactions aren't very, very strong. But despite this, there exists types of nucleic acid structures that are formed only through this specific binding of metal ions. And today, I would like to talk to you about a few of these systems. And one very, very well-known group are so-called G quadruplexes that were studied in the group of Professor Gdaniec since many, many years. And this is a kind of structure where up to 4 strands can associate into this kind of quadruple helix type of fold that is actually held together by different interactions than Watson-Crick base pairing, so-called Hoogstin base pairing, that I will talk to you about a little more in a moment. So first, I would like to present you one of the projects that we realized in our group related to G quadruplexes. We actually have many, many more data today on other G quadruplexes, but this is something I would like to save for future seminars. So this project is a little bit older, but at least it's complete and I can show you what we usually end up with. So an introduction to G quadruplexes is that these so-called G tetrads are an alternative to Watson-Crick base pairing. As you can see, once again, you have hydrogen bonds from one base to the other. However, usually it's this phase of the bond, of the base, the so-called Watson-Crick phase, that's making the interactions. Well, in this case, also this other phase, the so-called Hoogstin edge, is involved. That's why we call them Hoogstin base pairing. And as you can see, this is a homotetrad formed by 4 guanosine residues. And what's very important is that we recognize the formation of either the tetrads or base pairs by the observation in NMR of the iminoprotons of the nucleobases. When a nucleobase is unpaired, you don't see these protons because they exchange with solvent very, very fast, while when either form of base pairs or base tetrads are formed, you can start to see those protons because they are protected from exchange by hydrogen bonding. And this is very, very important because For us, this is usually the main thing based on which we evaluate what kind of secondary structure is forming. And actually, you can differentiate these two because iminoprotons in base pairs have different chemical shifts than iminoprotons in G tetrads. This is going to be important later. The point to keep in mind is that if we see these protons, we know that the given base is involved in interactions. If we don't see them, it probably isn't, or at least the interaction isn't stable. But getting back to the subject of G quadruplexes are formed by stacking of several such G tetrads, similarly to how duplexes or double helices are formed by stacking of base pairs. However, there is one very important difference. The formation of G quadruplexes requires metal ions between the quartets. And this is because the formation of Hoxton base pairing puts these 4 carbonyl oxygens very, very close to each other, and each one of them carries a partial negative charge. This is not a full negative charge, a partial one, but we have 4 of them very, very close together. And actually, if you have 2 tetrads, it's 8 because it's 4 from here and 4 from here. So in order to this not to dissociate immediately, we need a positive charge in the middle to shield these interactions between negative ions. Between negative charges. That's why positive ions are absolutely necessary for the formation of G quadruplexes. But G quadruplexes actually go beyond just G tetrads. Actually, no other nucleobase is able to form such stable tetrads as guanosines. So we only have G quadruplexes. We don't have C quadruplexes or U quadruplexes. However, when there's already a scaffold of several G tetrads that puts other bases in a conformation very, very favorable to form a tetrad, also other kind of tetrads can form. And in quadruplex structures, we already observed formation of uridine tetrads, adenine tetrads, cytidine tetrads, and all kinds of mixed tetrads. If you have this scaffold of G tetrads, then also many other type of four-stranded interactions can form. And the one project I would like to present to you today is one where we studied a G quadruplex formed by the sequence UGG, UGG, U, that actually in its structure, apart from 4 G tetrads showed here in yellow, contained 3 uridine tetrads in 3 very different chemical environments, at the 5' end, and the 3' end, and in the middle stacked between 2 G tetrads. And what was very interesting about this system was that it was unexpectedly thermally stable. Here you can see the spectrum where we see these iminoprotons, and I told you that if we see them, then there are interactions. If the structure will be melted, these iminoprotons would disappear. And you can see that they remain protected by hydrogen bonds even at 80 degrees, probably even higher, because we saw that the melting temperature of this quadruplex is around 90 degrees. And we weren't actually the only people that saw that if you put a uridine tetrad specifically at the 3' end of a biomolecule or of a G quadruplex, you see a huge increase in thermal stability. This G quadruplex without this U tetrad melts at around 50 degrees, while with this U tetrad, around 80. If you put the U tetrad in the middle or on the 5' end, you don't see this effect. So there is something very special about this uridine tetrad at 3' end, and we were very interested to find out what. So of course we did NMR spectroscopy study, and I would just like to show you the 1D spectrum because it's so deceivably simple. This is a 10 kilodalton biomolecule, and the spectrum looks more like a small molecule. You can see that the lines are a bit sharper compared to these lines here that are the products of degradation into nucleotides. You can see that the lines of the G quadruplex are somewhat sharper, but if you compare them to proteins, it's actually nothing. They are very, very few, and this is because this molecule actually has a 4-fold symmetry. So all 4 of these guanosines will give you degenerate signals, or 4 of these guanosines will give you degenerate signals, and so on. And this simplifies the spectrum greatly, but on the other hand, you will see that it was quite a challenge for structure determination. One other important thing that we saw already in the 1D spectrum and actually confirmed what kind of proton it was by 2D experiments was an additional 8 resonance in the aromatic range. And actually, by additional experiments, we were able to say that this is not an aromatic proton. This is actually an OH proton. Each single of these nucleotides contain a 2' OH group, but most of them are invisible because they exchange. And usually you don't see OH groups in RNA. This is one very, very rare example where we can actually see an OH group, which suggested that something strange happens structurally that leads to its protection from exchange. So to find out what was this thing that was responsible for both the protection of this proton and the huge stabilizing effect, we of course decided to solve the structure. And just to tell you briefly how we do it, NMR structures are usually solved by the means of molecular dynamic simulations restrained using NMR restraints. And the most commonly types of restraints are distance restraints from NOE. So we measure hundreds upon hundreds of distance between pairs of atoms, unfortunately only observable for very short distances. And the second type are dihedral angle restraints from J couplings. And in this very specific system, the NOE part was actually quite tricky because all of these NOEs were, as we call them, ambiguous restraints. Because there were 4 strands that had the same chemical shift, we never knew if a given contact, for example, between an atom from residue 5 to residue 6, was it within the same RNA strand, or was it from one strand to one of the 3 others? That's why we had to treat all of these NOEs as ambiguous, and this gave us a lot of local minima during the simulations. So the simulations are usually performed by the so-called simulated annealing protocol, which just means that we heat the system to very high temperatures to allow it to jump through energy barriers very, very efficiently, and then slowly cool it to hopefully let it fall down into the global minimum that is the structure that satisfies all the restraints. And for nucleic acids, we usually do it in a 2-step procedure. So in the first step, we start from completely unfolded 4 strands. Then we fold it using a simplified force field to get more or less the 3D shape we want. And then we do refinement with a more with a higher quality force field just to find the exact geometry. And this was especially important in this case where we had ambiguous restraints, because there were so many local minima that the simplification of the force field in the first step was very, very necessary to even be able to move from here to here. And the refinement later allowed us to get a very, very nicely defined structural bundle. And as you can see, there is a very funny, very peculiar feature in this structure. Throughout all the molecule, the backbone appears to follow a more or less helical path until the 3' end, where it suddenly shifts. This is between the residue 6 and 7, where you can see this strange hook that we did not expect. If you look at it at an atomistic view, you can see that you have the tetrad from G6 and tetrad from U7, and there is a very, very sharp turn between them. And actually, the final tetrad forms, but it is flip on its head. Usually you would have it on this side, while it's stacking with the other face on the previous guanosine. That's why we call this new structural motif a reversed U tetrad. And we believe that if we found a new unexpected structural motif here, this is probably the motif that's responsible for the thermal stabilization, but we just solving the structure didn't know how. We had to somehow go for deeper studies to be able to discern what are the sources of this thermal stabilization. And the first thing already presented itself very, very early from the structure. Namely, we've seen that the OH group that I mentioned earlier actually forms an additional hydrogen bond to the phosphate group of the previous residue. Usually this wouldn't be possible because this OH group would be somewhere here facing the solution in a normal conformation. But due to this flipping, we can have this kind of nice hooking effect. And actually these are 4 of these hydrogen bonds because it happens in each 4 of the strands. We kept analyzing, and it very nicely explains why do we see this OH group. So we kept analyzing the structure. We made additional analysis of the stacking interaction, additional MD simulations, which found that actually this accumulation of atoms in a very narrow piece of space actually forms additional potassium binding sites in the grooves, but just observing these additional interactions doesn't tell us that they are actually responsible for the stabilization. So what we did, we actually introduced a series of chemical modifications into this last tetrad. So we, for example, switched ribouridine to the oxyuridine, or to a uridine without the OH group, or to a citidine. And for each of these modified quadruplexes, we once again did the full NMR analysis to find out if the structure of the motif is preserved. And at the same time, we did thermodynamic analysis to see if the modification changes the thermal stability. And I won't bother you with details, but what we found out is that we are actually able to say that two things play the key role in this additional stabilization of the G quadruplex. And these are the additional hydrogen bonds I mentioned before, and the favorable stacking interactions. Actually, stacking of uridine on this other phase is in a better geometry than standard stacking. And these are the two things that are most important for the stabilization of the G quadruplex. So just to give you some conclusions, this is the kind of results we can hope to get from studying quadruplex structures. In this case, we were able to find the minute structural details that are responsible for a very pronounced physical effect, which is the thermal stabilization of this fold. And this was the example I wanted to show you about quadruplexes. There are also other non-canonical nucleic acid structures that are reliant on specific binding of metal ions. And these are the spatial structures formed by the so-called multiple way junction motifs. Multiple way junctions are DNA or RNA structures where you have helical elements based on standard Watson-Crick base pairs. Here you have 2, here you have 3, but they are connected by what you might consider to be flexible linkers. However, in the presence of an additional molecule, like a metal ion, it's possible that these flexible linkers will structure, forming some non-canonical element, and allow the whole motif to assume a single tertiary fold. And these are the two systems I would like to talk to you today. One of them is an aptamer binding lanthanide ions and forming a very, very intricate tertiary structure upon it. And the other one is a DNA zyme molecule, which binds many different ions. We studied with zinc, which also forms a very intricate fold upon metal binding. But just to explain the two terms I used, so aptamer for this system and DNA zyme for this, aptamers are actually DNA or RNA molecules that are characterized by one thing. They are able to selectively bind a chosen molecular target. So an aptamer is not a type of structural motif, it's a functional domain. Something is an aptamer if it can specifically bind a chosen molecular target. And these targets can be very different. There are aptamers for small molecules, like some organic aromatic molecules. There are aptamers for proteins that are able to recognize a specific protein among many others. And there are also aptamers for metal ions. And this is one I will show you today. While DNA zymes are in turn DNA molecules that are able to catalyze a specific chemical reaction. Many of these reactions are related to nucleic acid processing. So for example, it can catalyze RNA cleavage, it can catalyze RNA ligation, it can catalyze DNA phosphorylation, but there are also more exotic examples. For example, it can even catalyze carbon-carbon bond formation by this other type reaction. And what is actually very, very important about both of these types of systems, that DNA aptamers and DNA zymes were actually not yet observed in nature. The funny thing is that their RNA counterparts were, but DNA never. And despite this, we somehow have a lot of these systems, which means that they must have been made by humans. But they were made in a very indirect method, let's say. They were not designed because we don't yet know, we don't have enough structural knowledge to design a DNA molecule that binds a specific target. They were instead not designed, but selected. Basically, all the aptamers in DNA enzymes we know come from the process of so-called in vitro selection, or SELEX, which works in a way that we start with a random sequence of, a random library of DNA sequences, many of them, billions upon billions. Then we incubate with a given target, and we are able to select those that bound to the target and throw out those that didn't. However, usually this pool is still contaminated by many inactive sequences that just came for the ride. That's why the library is regenerated by PCR amplification, and the process is repeated. Often it's repeated many times, and in the end, you often end up with just a couple dozen DNA sequences coming from these billions upon billions that are able to bind the specific target. And so the outcome you get from SELEX is a set of DNA sequences that can do a specific task, catalyze a reaction or binds a specific target. You can get some information what residues are important for this, because if you have, let's say, 100 structures, you can see which stretches of residues repeat. However, you don't get any information regarding how this molecule actually works or what structure it assumes. That's why these kind of systems are great systems for structural biology characterization. I don't know whether you can actually say biology, because these are biological macromolecules, but let's say designed by humans. However, the point still stands that these are most often very uncanonical, very rare DNA structural motifs. Just think about it, that it's just a couple dozen structures selected from billions upon billions. So they have to be very rare types of faults. And we actually don't know anything about them, while at the same time they're very important due to their action. And I would like to show you two examples of this kind of systems. First of them is an aptamer that was discovered in 2016 that is able to bind lanthanide ions. And actually what was very special about it is that it was able to bind these lanthanides with very good affinity, very rarely achieved for nucleic acids, sub-micromolar affinity. And what they did in the original paper, this was the library in this SELEX experiment. The ones in yellow are the random parts, and these are the 22 sequences they were able to find. So this part is these 20 random residues. They were able to see that there is actually some base conservation within. They were able to propose a secondary structure for this aptamer, and even seeing that this important part and the conserved part was all in this loop, they were able to cleave many parts this bottom. They were able to make this helix shorter and arrive at an aptamer like this. They knew that the lanthanide binding site was probably somewhere in this loop, but without deeper structural studies, there was no information where exactly within this loop the ion binds, or what is the geometry of the binding site. And this is where we enter. We decided to study this by NMR. However, as we usually do NMR without isotope enrichment, it's very important for us that our system that we study is the smallest possible. That's why, looking at these sequences I showed you before, we worked out a tentative consensus structure for this aptamer. And we've seen that you can probably do a lot of things to the arms. You can probably change this part of the loop. What's important is that you conserve this and this. And through trial and error, trying many different DNA constructs, we arrived at a shortened construct that was still being able to bind lanthanide ions just as well as the long one, but it was only 28 nucleotides in length. And this is the, let's say, NMR optimized construct on which we then did structure determination. And one thing we introduced in order to be able to stabilize a very short helical element, we used a so-called GAA tree loop that is a most stable trinucleotide loop in DNA. So if we put it on a short helix, it actually stabilizes it. And it's also very interesting from NMR because it forms a shared GA base pair. So these two bases actually pair. and it has very unique chemical shifts, so it's very easy to spot it in an NMR spectrum. So just to show you how the 1D spectrum of this system looked like, once again, this is just the imino region, so where the base pairs are seen, and the aromatic region. And you can see without metal, there are already some base pairs. You can count 5, maybe a 6th one. So more or less the amount you expected here, even slightly less actually. But when you add one single equivalent of the metal, the spectrum changes completely. You can see that many additional immunoresonances appear, and basically everything becomes sharper, probably because some exchange phenomena that were initially present are now no longer in the system. It assumed a single tertiary fold. And of course, to assign the spectra, we needed to do a 2D analysis, then extract the restraints and so on. But even without calculating the structure, just upon the assignment of resonances, we were actually able to say that the secondary structure of this aptamer is actually more complicated than what you can see here. Namely, during the assignment of 2D spectra in the presence of lutetium, we observed additional structural elements that were not predicted originally. We've seen a so-called wobble GT base pair, so a pair between guanosine and thymidine. Next to it, an additional GC base pair that was not in the original secondary structure. And actually, I told you that the GAA3 loop has a very specific NMR signature, and we were very surprised that we saw it in the spectra twice instead of just once. We introduced this GAA loop, but actually we've seen 2 GAA loops. And the only explanation for an additional GT, GC, and additional GAA was that actually the secondary structure of this aptamer is much more involved. All of this part of the loop actually forms a third helix. You have the GAA here, a GC pair, and a GT pair. So already without doing structural calculation, we've seen that this aptamer is actually much more structured in the presence of lutetium than initially anticipated. So the next thing we did was, of course, we extracted the structural restraints and calculated the structure. And the protocol in general terms is very similar to what I described to you before. However, there is one very important difference. We are able to use for this system a third type of structural restraints, so-called paramagnetic NMR restraints, that are very sought after because not like NOEs, they are very long-range restraints. So I'd like to tell you just a few words about these paramagnetic restraints, what they are, before we continue with the structure. So paramagnetic restraints are additional NMR observables that can be seen in the spectra if there is a paramagnetic ion within the biomolecule. And actually, for lanthanides, apart from lutetium and lanthanum that are on the edges of the series, 13 other lanthanides are all paramagnetic. So if we put several different paramagnetic lanthanides, we will see all of these effects. And these effects have their specific names: pseudo-contact shift, paramagnetic relaxation enhancement, or residual dipolar couplings. They show themselves in the spectra in different ways. PCS are shifts of resonances, PREs are broadening, and RDCs are differences line splitting. But in this specific project, we concentrated on PCS because before they were really not easily observed in DNA molecules, and we wanted to see how good actually the PCS will be from this lanthanide binding site. So just to give you a quick introduction to PCS restraints, they are long-range NMR restraints. They can be seen up to 30 or 40 angstroms from the unpaired electron. And what they do, they actually correlate the position of every single atom in the DNA with the position of the unpaired electron. So in other words, with the position of the metal ion. The mathematical expression that describes what will be the shift observed at a given position is quite involved, and it depends on the distance from the electron to the atom. It depends on the angles in the principal axis frame of the magnetic susceptibility tensor of this unpaired electron. However, I usually think that it's the most important thing is to stress that they are observable for very long distances. This is what distinguishes them from NOEs. Even though they are more difficult to analyze, they can give you information that NOEs can't. And just to simplify a little bit what this expression means, I would like to show you some visualization of this equation. So, for example, we can ask ourselves, this is the reference frame of the metal ion, and we can ask ourselves, what are all the points in space where we will see a shift of, for example, +1 ppm. So if you ask this question, you can draw a so-called isosurface. So all the atoms located on this red shape will feel the shift of +1 ppm, while all the atoms on this blue shape will feel the shift of -1 ppm. And then if you want to ask about other shifts, we've seen that there is this distance dependence. So if you see a smaller shift, you know you're on a similar shape, but bigger. If you see a negative shift, but smaller, you see that you're on this kind of shape, once again, but bigger. And the smaller the shape, the smaller the shift, let's say, bigger the distance from the unpaired electron. So this is the theory, but did we actually observe this effect for our aptamer? And yes, actually we did. When we titrated, instead of lutetium, other paramagnetic lantanides, so europium, ytterbium, or cerium, you could see that the chemical shifts of iminoprotons are very, very different. Before the last proton was here, in europium it moved quite a bit, in ytterbium it moved once again quite a bit. And to know which proton is which, you have to actually assign all of these spectra. But if you do this, you can actually extract the PCS restraints directly from the chemical shifts that you can see here. So for example, for europium, the chemical shift of iminoproton of residue T7 is like this, something like 0.7 ppm. For ytterbium, it's higher, and so on. And as you can see, in many cases, some metals give you shifts in one direction, while other metals in the other, because this principal axis frame, as I will show you, is different for every metal. But now let's look at the structure. What was actually the effect of using all of these intricate NMR methods? So this is the high resolution structure of our aptamer. And as you can see, it actually forms one tertiary fold. These two helices, the green one and the blue one, make so-called coaxial stacking. So they actually form one long helix by stacking one on top of the other. Well, this one is at around 90 degrees from them. And the metal ion is in the center of all of this. So if we look a bit closer at the metal binding site, we can see that it actually interacts directly with three groups from DNA, two phosphate groups, and a cytidine carbonyl atom. And actually, you can see this interaction directly in the phosphorus spectrum, because these two phosphates, upon binding the metal ion, are shifted very, very much by a few ppm from their original location. So actually, you have direct confirmation that these two phosphate groups interact directly with the metal ion. We then did some additional MD simulations to see if there is some specific hydration pattern around the metal, and it seems like there is, but this is not important for us today. What is important is how these PCS data helped us to actually better define the structure. So just to show you how we use this PCS data, this is the structure for our aptamer, and now we're looking at the data for europium. If we put the PCS values that we observed on the structure, so the red ones are positive PCS, the blue ones are negative, the white ones are around 0, then we can try to fit this to the equation I showed you before. And as you can see, the regions fit ideally into this kind of shape I showed you before. It's even better seen if we rotate the image a bit, that there is this part of the space where you can have negative PCS, part where you have positive ones, and between them, on this axis exactly, there is a point where there is exactly PCS of 0. And for every single metal, what you can do is see how the PCS observed experimentally compare with the one computed from the equation. And if you have this kind of good agreement, this means that exactly this one single structure is able to explain all the paramagnetic effects. This means that the structure is actually rigid and the metal is at a single position. And for each single metal, you will have a different pattern of this PCS. As you can see, for ytterbium, they were generally much stronger, but also oriented in a different matter. And for all metals that we used, we of course seen different patterns of PCS, but in every single case, they were very, very well reproduced by the structure. This means that this is actually indeed a stable tertiary structure, and the metal position is very, very well defined. And here I show you how the use of this PCS allowed us to better determine the structure. The blue bundle is the one that we deposited in the PDB, so the one that we determined with all the data, the NOEs, the PCS, all the restraints, while the purple one is the NMR bundle that we obtain if we don't use PCS. As you can see, it's much, much worse defined. And actually, if you compare to representative structures, they don't even fit. This is because when you don't use long-range restraints, such features as interhelical angles are very difficult to accurately model using NOEs, and actually the force fields can somehow bias what the angle is. So not only this structure is much more better defined, it's actually also more accurate. And we hope that we'll be able to now extract this binding site and move it to other DNA molecules to also use these PCS restraints in our future studies. But this is now work in progress. This is something I was hoping to be able to do for a very long time because I'm a huge fan of these paramagnetic restraints, but this is still work in progress. And let's now move to the last system I would like to present to you today. And this is the so-called 817 DNAzyme. This is an RNA cleaving DNAzyme, which means that it can recognize an RNA substrate. This is this strand at the top, bind to it, and then cleave it in a very specific place in the structure. What's, however, crucial, it cannot do it without binding a divalent metal ion cofactor. And it's not very choosy. It can bind a lot of metal ions. However, the problem is the main application we would like for this system is in, let's say, as a drug that can find a specific RNA sequence in cell and then cut it to decrease the expression of a given gene, for example, the gene that's overexpressed in cancer. And this DNAzyme was discovered in the 90s. And since the 90s, there were attempts to use it in this way. However, they were still not successful due to exactly the metal binding property. Namely, in the cells, you only have appreciable concentrations of magnesium out of all of these ions. And magnesium affinity for this system is actually very, very low. This means that this DNAzyme is not very active in cellular conditions. It does some RNA cleavage, but it's very, very suboptimal. That's why it was not possible to change it into a drug, even though for 20 plus years people were trying. So we would actually need to engineer better variants of this system. But as I told you before, for DNAzymes, we don't really know how they work. This is, as I remind you, because we do not design them, we select them. So we know that this sequence is a DNAzyme that cuts RNA, but why it does it? Well, this requires structural studies. And people tried it for many, many years without success. People also used lower resolution methods like FRET or CD spectroscopy. And actually these methods uncovered a very peculiar thing about this DNAzyme. Namely, they've seen that if you add ions such as magnesium or zinc, you see a structural effect. Here what you see is the FRET effect between probes put on these two helices. You can see that the FRET effect increases. So metal binding brings these two helices together. But what was very strange, binding of magnesium does it, that binding of zinc does it, but not binding of lead, which is also a cofactor. In CD spectroscopy arrived at a very similar conclusion. Zinc and magnesium changed the CD spectrum, while lead didn't, which meant that it seemed that this DNAzyme actually has two active forms. Let's say a more open one that's present without metal ion, and that lead binds directly to it, can activate it and cleave RNA, while other metal ions actually induce a different active state, a more compact one, with the arms closer together, and cleave RNA from this, which was very, very strange because it would mean that this is an enzyme that can work from two different active states. And for many, many years, the characterization of either state was not possible until in 2017, the crystal structure without metal ion and with lead was solved. It turned out to be much more intricate than anybody expected. It actually revealed the lead binding site. This part of the molecule here forms like a basket into which lead can slip, and then it's very, very close to the RNA cleavage site. These are just two guanosines I show you here. And in this paper, they were able to actually propose what is the catalytic mechanism for the very first time. And this was a huge step forward. However, still, based on these previous results, everybody thought that, okay, now we know the structure of the inactive apostate, we know the structure of the lead-bound state, but actually what's relevant for biology is the magnesium-bound state that we still do not know. That's why I decided to try to solve the structure of the system in the presence of zinc that was supposed to do the same as magnesium, just to put this final piece of the puzzle, what is actually this closed state induced by metal binding. And to do so, once again, we started with DNA construct optimization because this DNA enzyme is quite big. And we used two tricks. One of them was the truncation of the arms, but we had to increase the CG content to make them still want to fold. And we connected the two strands, once again by a GAA tree loop to form a unimolecular construct. We once again tested quite a variety of them, and in the end chose one that formed a very stable structure and actually had a great dispersion of NMR resonances. This is this immuno region. As you can see, there is barely any overlap. This rarely happens. So what we did actually to study the DNA enzyme, we constructed this kind of shortened NMR optimized construct. And first of all, we had to check whether it still works, and it did, and whether it shows metal-dependent folding. So by CD spectroscopy, we were able to see that it actually reacts to the presence of zinc with a binding constant similar to what was known from the literature. And we can also see a similar effect in the NMR spectrum. We've seen that some additional resonances rise when zinc is added, some others disappear, and actually already one equivalent of zinc basically saturates these changes. Additional equivalents don't do nothing. So it seemed that this is what we observe is indeed the compact zinc-induced structure. We, of course, had to assign it, and usually assignment in DAS is done based on NOE-based methods. We follow so-called nosy walks, so expected paths of connectivities between residues, but they are actually expected for helical regions. If we have strong non-canonical interactions, it might be that doing the assignment like this will actually fool us. That's why for Q residues, we also used site-specific carbon and nitrogen labeling, but this was quite work intense because for each one of these graphs, we actually had to make a new construct of the system. So for example, to assign the iminoproton of this guanosine, we needed a new construct when we labeled this guanosine. To assign the aromatic proton of this adenosine, we needed a new construct, and so on and so on. However, it was worth it because it was important to say that even in these unpaired regions, we can get accurate assignments. So once again, what we did, we took all the NOE data that we had and decided to solve the structure. This time, unfortunately, no paramagnetic effects. And what we arrived at was once again a very interesting, intricate structure. We've seen that actually, apart from the 3 helices that were expected, there was also a 4th one formed by one non-canonical base pair, GA and GC. And this kind of structure is actually called a pseudoknot. And the whole tertiary structure is what actually, once again, a very nicely defined one. However, one thing that came as a shock to us was that when we superimposed it on the crystal structure, they were actually basically perfectly superimposable. The crystal structure, of course, had longer arms, but this is just due to our construct optimization. The catalytic domain that's in the middle is basically the same between the lead-induced crystal structure and our zinc-induced NMR one, which is completely against all the previous literature. That's why we were quite shocked, quite puzzled, and actually solving the structure was only the first step to explain what is happening. So what we did, we actually checked whether other metal ions perform as we expected from the literature, and we see for both sodium and magnesium that actually they do fold the DNA enzyme. What came as another shock is that when we tried to use lead, also lead made more or less the same spectral changes as the other 2 ions, which is something that nobody ever seen before. But as we had now the samples stabilized by each of these metal ions, we of course studied them by 2D NMR, Not all of the spectra were of such a great quality as the original one, but we are able to assign all of them. And while we didn't solve the structures anew, we are able to compare the chemical shifts with the original conditions and also the NOE patterns. And we were able to conclude that basically the chemical shifts and NOE contact patterns are very similar whatever ion we add. So actually, we're even more confused because everybody said that lead doesn't fold this DNA enzyme, but at NMR concentrations, what we saw was actually that lead was no different from all the other metal ions. And we will come back to this in a moment. Because there is one other thing that this conclusion allowed us to do. Because now we know that both sodium and zinc actually induce the same structure, but actually sodium has a much lower affinity than zinc. Zinc is micromolar, sodium is millimolar. And this allows us to try to study the zinc binding site using chemical shift perturbations. Because usually what you do when you want to study interactions with something that cannot give you NOEs like a metal ion, you just titrate it and see which chemical shifts in the molecule are changed upon metal binding. However, this relies on the assumption that the metal binding doesn't change the structure, while in this case, this is clearly not true. However, if we are able to pre-structure the DNA enzyme with sodium and then titrate with zinc, zinc will simply eject sodium without changing the structure, and then all the chemical shift perturbations should be very close to the zinc binding site. So we did exactly that. We titrated zinc, but this time to a DNA enzyme pre-folded by sodium. We're actually able to see a limited amount of chemical shift changes, just to a few residues in a very well-defined spot. And if we map these chemical shift changes on the structure of the DNA enzyme, we can see that they are all in one part of the structure, which we can assume is the zinc binding site. Unfortunately, the CSP method cannot give you the exact geometry of this site. What was shocking, however, is that the phosphate that is cleaved showed almost no CSP, as previous literature showed us that actually this phosphate should bind directly to the metal. As you've seen before, direct coordination to the metal in case of the lanthanide aptamer gave us a huge shift, while here the shift is almost zero. That's why we started to wonder, is the metal binding we observe actually the one relevant for catalysis? Because the metal binding, the zinc binding that structures the DNA enzyme, this is a micromolar site, while if you monitor zinc binding by activity, you actually always get a millimolar affinity. And we were not the first people who observed it. There was one paper in 2007 that showed the same, but somehow it was never followed up after. But it seems like there might be more than one metal binding site in this system, one related to the structural effect and one related to actually activating the DNA enzyme. And this actually somehow explains our CSP results, that we don't see any CSP on the phosphate, because actually we observe the binding to the strong structural site. And I don't want, I know my time is running up, so I don't want to show you many more experiments, but starting from this idea and doing additional experiments, we're able to propose a new idea how actually this DNA enzyme interacts with metal ions. So this is the classic model that I showed you before. What we propose instead is that the apoform is not actually a single state. It's a mixture of different conformations. As we've seen when we titrated metal ions, there were some forms that disappeared. And in the apoform, you have some fraction of the well-folded state and other states that are inactive. And zinc, that binds very well to the structural site, first thing it does, it structures the entire population of the DNA enzyme, and only when you add additional zinc, it starts RNA cleavage. While for lead, we observed very similar binding constant to both the structural and catalytic side. This means that lead actually activates the DNA enzyme without fully changing the conformational equilibrium. Only additional equivalents of lead move the whole population of DNA enzymes into this 100% folded state. And this is the reason why previous experiments observed folding with zinc, and not with lead, because previous experiments always used concentrations of zinc that actually activate the DNA enzyme, so they passed right here, and concentrations of lead that activate DNA enzyme, so they just ended up here. So our new interpretation is that there is no two active structures, but rather two metal binding sites that are responsible for this different behavior of lead. So just to summarize, we're able to solve structures of three very, very different DNA and RNA molecules that are related to each other by the fact that they require specific binding of metal ions to form. These are the publications we were able to put our results in. I just show them so that if you want, you can check them out later. And this is just part of all the research work of all the projects that we do at NMR Group. But I think there will be future seminars, so I will be able to tell you about other projects that we do. And in the end, I would like to thank all the research group, of course, Professor Zefjak Daniec, but also all the other people that were involved in this research. And of course, thanks to you for your attention.
Thank you very much for your presentation. And I will remember that you have more topics to talk later. Okay, so we are open for the discussion. If you want to ask the question, please turn on your microphone and camera. If you have problems with microphone, write it on chat.
Hello, I have a question. Yeah, thank you very much. It was very nice talk. I have maybe 3 questions. So first of all, which force field have you used in your MD? Second of all, what would you say would be limitations of PCS to RNA systems? Will it work to all of them? And the third, when you showed that you had the structure of your RNA, which was longer, I think 40 nucleotides, and then you shortened, then you say that you see differences like the 3 loops, etc., than what expected. How can you, on which base can you say that the differences are not because of the shortening of the structure?
Okay. So if I remember correctly, the first question was the force field. So for nucleic acids, we almost exclusively use amber type force fields, because for proteins, it's usually, well, the other one. What's the other one apart from amber? Anyway, for nucleic acids, we use amber force fields. They are much better validated for this kind of systems. And when it comes to the PCS limitations, actually, well, we do not know what is the exact range of limitations, because right now, the main limitation is that there are very, very few ways we can induce this kind of restraints for DNA and RNA systems. For proteins, there was a lot of work put by many research groups to develop so-called lanthanide binding tags. So either small molecules that you can covalently attach to a protein, or some protein loops that bind lanthanides. And now you can use the PCS and RDCs and PREs very, very easily just by attaching this tag. While for RNA and DNA, this work wasn't done yet. Actually, the system that I present you is, as far as I know, the first system where we're able to see measurable PCS, and not only measurable, but also interpretable, for a direct interaction between a metal ion and a DNA system. By the way, this is DNA, not RNA, this aptamer. So the main limitation for now is that we don't have proper methods to induce them. When we will have, then we can think about what are the other limitations. So this is the main thing I actually want to bring to the table, methods to induce PCS data for nucleic acid systems. And when it comes to the last question, I will try to get back there. You are probably referring to this slide here, right?
Yes.
Okay. So the main thing, what makes this DNA molecule an aptamer is not its structure. What makes it an aptamer is the ability to bind lanthanide ions. So what we were monitoring during this process is whether lanthanide binding ion, whether the binding of lanthanide ions is affected or not. If we made a change and suddenly the binding constant was worse, we knew, okay, we messed up something in the structure, we cannot use this construct. However, this construct that we got has actually metal binding constant even slightly better than the original. So we know that we didn't mess up anything important in the structure.
So because the function is preserved, but maybe in other
what defines the
systems
aptamer is its function.
Okay, thank you.
Anyone else? If not, maybe I'll ask a very naive question. So I was just curious about the SELEX method. Are there any attempts to replace it with some tools similar to AlphaFold, where you basically predict the sequence of your interest?
This would be great, but AlphaFold works because we had this 100,000 protein structures that we could train it on. For aptamer structures, we have, I won't give you exact number, but I think around 100 probably. So,
okay, so
we're
not
far away. And the aptamers are very, very diverse, because aptamers for proteins, aptamers for small molecules, for metal ions, you would probably need orders of magnitude more structures to find out what features in DNA allow it to interact with a specific target. So we are, I would say, ages away from being able to design them. The SELEX method itself, it's like 30 years old, and there are still improvements made to the method itself, but the main point still stands, that it gives you a DNA or RNA that can do something, but you do not know why it does this thing.
So I have a question, if I can.
Yes, of course.
Okay. Also about the selection, you said that you have some kind of DNA library which contains like 10 to the power of 14 molecules. How do you come up with them? So where do you take them from? Because this is a very large number, and and you have to have them physically, right? You really use them.
Yes, and if you really think about it, if you have this library in a, let's say, micromolar quantity, there are just a few copies of each of these molecules. So how do you even get this? And the way you do this is, the funniest thing is that if you have, let's say, a random region of, let's say, 20, 30 nucleotides, and at each position you can have 4 different nucleotides, then the amount of structure is 4 to the length of the region, which is actually usually more than 10 to the 14th. So you don't have, let's say, a full sampling of all the of all the sequences. And how you get it is that usually we get our samples not from biological ways like in vitro transcription. We get them from chemical synthesis. And what you do to get this kind of random library, this is a synthesis similar to peptide synthesis. So what you do, you synthesize part of the DNA, and then when you want to add the additional bit, you give it a mixture of 4 nucleotides. So in your synthesis, you get basically sum of each of the sequence that can possibly form.
Okay, so you have to just stop the synthesis when you have a proper length, more or less.
Yes.
And so individual molecules vary in length, right?
No,
or they are
no, because the synthesis actually it's a cycle where you can ensure that at every cycle only a single nucleotide.
Okay.
So you add 1 nucleotide, you have to do some additional reaction to reactivate it again.
Okay.
Add 1 nucleotide, and so on. So usually these libraries look like this, that you can have some fragment that is actually well defined in sequence. Then you add some amount of random nucleotides. You can actually put once again some well defined ones, some random ones, and so on. Usually you do this because you want a specific secondary structure. For example, for the beginning of this to form base pairs with the end, so that in the end you will have secondary structures like this, that you have a stem, and then this whole random region is, let's say, up here. So what they were aiming for, this was the stem that was actually designed, and here was the long random region. What they found out, that most of the sequences that they found actually chose nucleobases in a way to form a long helix here and a loop here. So this is the whole DNA that they had, and these yellow parts were selected. So basically this helix was not planned by them. It just showed up because the process selected nucleotides that were exactly base pairing to this part here. So this is a very powerful method. You don't have to know anything about how to get the two molecules to interact. But in the end, the thing is that, well, you also don't know why they interact.
Yeah, but you know already what interacts, so I think it's already quite a lot.
In a way, because I hope that I showed you that, well, the structure that we observed was much, much more than this original secondary structure made us believe. We thought that, okay, this is probably a loop, lanthanides comes from somewhere here, and the nucleotides just, let's say, wrap around it. Well, in reality, we've seen that this is a much more intricate tertiary structure.
Okay, thank you.
Okay, I think we'll out a little bit of time, so maybe we stop the discussion here, and I just present the plans for the next seminar. On our next seminar, a moment. Yeah, we'll have Dr. habilitowana Marta Dudek from Center of Molecular and Macromolecular Studies for Polish Academy Science Łódź, and she will talk about NMR crystallography of organic solids at high magnetic fields when resolution matters. We have a full lineup till the summer break, and I'm also making slowly the list for fall. So if you want to have a presentation in fall, please email me. And thank you very much for participating today, and see you all in 2 weeks. Thank you very much.
Thank you.
Thank you. Bye.